are thrilled to have with us some of the experts in diagnostics that are going on in the state of Utah. And we're going to show off just a little bit how innovation is changing the landscape in diagnostic development right here in the state of Utah. There have been dramatic changes in laboratory medicine that are driving better health outcomes and shaping the lab of the future. We're very fortunate today to have Dr. Greg Critchfield moderate the panel discussion. He's a board-certified physician in clinical pathology. He became a director of clinical pathology for Intermountain Health and then served as chief medical and science officer at Co-diagnostics. Greg then joined Myriad Genetics, where as president of Myriad Genetic Laboratories, he built it into one of the world's leading molecular diagnostic companies. After Myriad, he became chairman and CEO of Sera Prognostics, where his team developed a pipeline of innovative diagnostic tests for the early prediction of preterm birth risk and other pregnancy complications. For Greg, innovation means being bold. His research is focused on building quantitative models in multiple areas of laboratory medicine. He received his bachelor's degree in microbiology from BYU, an MD degree from the University of Utah, as well as a master's degree in biophysical sciences at the University of Minnesota. Very inspiring and impressive. Please join me in welcoming Greg and his panel, and we will turn it over to them.
Thank you very much, Kelvin. It's really great to be here today. What I think we can do is give you some insights into some of the considerations that take place when you're developing new diagnostics. I go back to a comment made by Francis Collins years ago. This is back when he was leading the Human Genome Project and then became director of the NIH. He made this statement: genetics is medicine, and medicine will become genetics. Consistent with that statement, and as we've seen also by extension in practice, diagnostics is a key part of modern medicine. Modern medicine is largely becoming treatment based on diagnostics. You think of the number of diagnostic tests, the number 70% has been thrown around. That number actually came from Intermountain Healthcare. I had a conversation with Larry Grandia, who was head of all informatics at Intermountain years ago, and asked him, if you look at your records, what percentage of tests are informed by diagnostics? And diagnostics included radiologic exams and laboratory. Back then it was 70%. It's higher today, let me tell you. So really what Francis Collins said is that diagnostic information today, it informs healthcare workers about what biology is actually operative in a patient and what therapies are going to be useful in actually addressing the biology that's occurring. Today's panel is composed of very accomplished diagnostics leaders. They have broad and very deep experience. A lot of different things, but there are a lot of themes that we'll run through that you'll pick up on as we get into things. And I think it'll be really fun. I'm going to ask each of the participants to actually introduce themselves, say a bit about their companies, and after we've gone through the list of companies and you'll hear their backgrounds, then we'll open it up for questions at that time. So I'm going to go alphabetically just to be very fair for everybody. We'll start with Jared Bauer. We'll go last name first. Here you go, Jared. Would you mind?
Happy to. You just want me to introduce myself and the company?
Well, and if you have any presentation remarks you'd like to show, you can do that.
Yeah, there were slides. I don't know if they have those ready.
I don't know what order they're in, actually.
Oh, look, that's me. Okay.
Hey,
that worked out really well. So I'm Jared Bauer. I am the CEO and co-founder of Seek Labs. Seek Labs is a local diagnostic company. We've actually been working on our technology for 7 years. Our goal is to move diagnostics, frankly, into the home. We really believe in this idea of patients receiving their answers. Point-of-care diagnostics is a term that gets used by a lot of different people for a lot of different things, but for us, this actually means the whole breadth of wherever the patient is. So if the patient's in a clinic or hospital, we view that as point of care. If the patient is at home, we view that as point of care. Or even if the patient is in a rural village on the other side of the world, that for that particular patient is point of care. Now, this device was on my seat, so this also maybe seems to be working out. Down. Well, I appreciate that. Any other ideas?
Yeah, can we get some help with the...
I want you to know that I run a tech company, and I'm just so excited that I can prove my skill set here. Maybe if I point it at the sky. Point it over there. Yeah, I'm doing that too. Hey, you know what? I'm really proud of our logo, and let me tell you what it means. I'm gonna hand this off. This feels really redeeming, actually. So I'll tell you what, we have some great slides for you today. We're not going to show those. Instead, let me tell you what we have. So for us, we've been at this for 7 years, and over the course of the last 7 years, we've been really focused on developing a technology that could reach the patients at the point of care. And it's not. Oh, hey, I'd like to introduce to you the Seek-It system. I don't know how many times I can change on the fly, guys. We're running at the end of my skill set. So a week ago today, actually, we introduced to the world finally what we've been working on for the last 7 years, and that is the Seek-It system, or the Seek-It molecular diagnostic testing platform. And this device was actually created to run the full process of what we would think of in a molecular diagnostic test. So DNA, RNA extraction, amplification, detection, and communication. The goal was, from the very beginning, to follow the World Health Organization's reassured criteria. And I don't know how many of you are familiar with that, but the World Health Organization said this is the criteria that would really need to be met for a point-of-care device. Well, one of those elements, or a few of those elements, are actually incredibly difficult to overcome, one of them being the elimination entirely of equipment. You also have to have a rapid test. It needs to be sensitive, specific. Otherwise said, it needs to be a very accurate test. And that's something we really believe in. And so our device actually runs the full process and is very simple to use. I'm actually going to set this down. Let's try and balance it on my leg. Sorry, I should maybe, Dr. George, will you just, thank you. Appreciate the assistance. This is actually the Seek-It device right here. So start to finish. If there were cameras, I could show you what it really is. But this upper chamber actually does full DNA or RNA extraction. We then have an amplification chamber. We then are able to detect actually what's occurred. What's cool about this is that we've moved past traditional methodologies. So we've eliminated things like the silica-based method for DNA, RNA extraction, and we've eliminated the need for heat or temperature controls for our amplification technology. Allows us to develop tests for a broad range of sample types, including blood, saliva, swab, and tissue. And ultimately, with this device, we're actually able to give both a digital or an analog result, which gives us really the ability to enter into multiple markets. In the United States and in Europe, and frankly in a good chunk of the world, we really do need a digital result because we need to integrate into laboratory information systems and into electronic medical records. But there's another about 6.5 billion people where that maybe isn't what is needed, and so making sure that we have an analog result for those markets. So that is Seek-It, and that is something, Seek Labs, we've been working on it for 7 years. We didn't even have a website till about 45 days ago, and that is the Seek-It system released last week.
Very good, thank you. Let's go next to Kyle Berg.
Nice to meet you all, and I'm so excited to be here today. So Kyle Berg, Quansis is what I titled the slides. Okay. So a little bit about me. My name's Kyle Berg, and I am one of the luckiest people in the world because after I did my master's and bachelor's here at Utah State in the state, and left to do my PhD at UCSD, I got to come back not only to Utah, but I got to come back to the most beautiful place in Utah, which is Cache Valley. My house backyard looks up into the Wellsville Mountains, and it's majestic. Tell you a little bit about our company. So Quansis Biosciences is a multiplex ELISA company. As you may or may not be able to see those small dots inside of each of those wells, but each dot would represent an individual ELISA assay that could be completed simultaneously with up to 17 other assays from a single well with a single sample. And that is really the power of our platform, is that we can take these ELISAs, put them together and generate enormous amounts of data, and then we have a software that will then go and analyze each spot individually and give you the results of 18 ELISAs from one without compromising performance. I'm a little bit different than I think a lot of the people up here in that we only deal with a narrow part of the diagnostic development pipeline. Really, we only fit into this first part of feasibility, development, and manufacture. And so that is kind of our niche. Quansis primarily and historically has been an RUO, or research use only, manufacturer. However, we're finding more and more that we are getting pulled into the diagnostic space as we build our kits following the M10 guidelines from the FDA, and these kits can then be used as an LDT by other companies that like reference labs like ARUP. One development that we are particularly proud of that we've completed recently is our Qplex human pneumococcal IgG panel. So. For those of you that don't know, pneumonia, at least bacterial pneumonia, is caused by a host of different serotypes of this bacteria. And we've developed a kit that is really helpful in identifying vaccine response, or I guess the immune response to vaccines in patients. And so you can kind of see down here below, you've got these kits are serological kits, so we're not actually detecting pneumonia, we are detecting an immune response to pneumonia. We're measuring IgGs in the blood. And it matches a lot of the vaccines that are out there in the market today. This is an extremely rapid kit, which is an advantage that we have over some of our competitors. This is an extremely fast ELISA with 2 30-minute incubation steps, a 5-minute read step, and from start to finish, that gets it done in about an hour and a half. No conjugations are required. All of that is taken care of in the design of the kits and the software that analyzes each of those spots. And it's referenced to the World Health Organization standards. And you can kind of see an image of how the design of that 9-plex is in that well, and how you get different intensities based on the response of the individual to each of those individual serotypes. The power in these kits really comes from the ability to take a large set of data and to draw inferences. For example, you can notice if we've got 9 different patients that were vaccinated, we've got a sample from each patient before vaccination and another sample after vaccination. And you can see that some patients are very good responders. They would not be immunocompromised individuals, but you can notice, for example, patient number 5 on my list here didn't have a particularly strong response, may or may not be an immunocompromised individual. From a vaccine development standpoint, if you take a look at the serotypes along the top, you can see that of the serotypes here, patients tend not to respond as well to serotype 12F. And that's just a piece of information that they could then use to move forward in designing their vaccines and that sort of endeavor. And that's it for me.
Okay, thank you, Kyle. Let's next go to Tracy George.
Hi, I'm Tracy George, and no slides. I'm president and chief scientific officer at ARUP Laboratories, and I'm a professor of pathology at the University of Utah Medical School. So I wear a lot of different hats, and I'm delighted to tell you about ARUP. So ARUP spun out of the University of Utah's Department of Pathology back in 1984. Now fast forward to 2023, and we're the largest nonprofit clinical reference lab in the United States. We have almost 5,000 employees, and we're located here in Salt Lake City. We serve all 50 states. We serve large healthcare systems as their reference laboratory. We serve most of the academic, teaching, and children's hospitals in the nations. And we do lots of obscure, esoteric tests. And that's really what we excel at. And we also have a whole innovation unit, and that's what I get to spend my time doing, and it's a ton of fun. So I'm in charge of our research and development, which is really a development team of over 100 scientists, and we support all of our production labs because we get over 85,000 samples a day. So that's a lot to manage, and we've got a lot of automation to deal with that. We also have a newly launched Research and Innovation Institute, and we're trying to kick off innovation for the laboratory and diagnostics. It's more about 5 to 10 years out, as opposed to my development team, which is 1 to 2 years. And then we also have a clinical trials and pharma unit, which I used to run, which is a lot of fun. We do a lot of companion diagnostics. Not surprisingly, we're a huge LDT laboratory developed test shop as well. I've got a fantastic job, an amazing team. ARUP is over 100 medical directors, and most of us are actually faculty at the U. And so we have a lot of deep expertise, and we've helped out a lot of local startups because they've partnered with us, and I'll be delighted, I think, in the question session to talk more about some examples. So nice to be here.
Okay, thank you very much. Let's next go to Andy Hemmert. Andy, go ahead.
Perfect. Good afternoon, and thank you so much. It's a pleasure to be here. My name is Andy Hemmert. I'm the head of molecular R&D for bioMérieux. Now, bioMérieux might not be a very household name. You've heard our name mentioned other times today already, whether it's BioFire or Idaho Technologies. The company grew over time and was acquired. So currently today, we are called bioMérieux. We have more than 3,500 employees in the state of Utah. According to BioUtah, we're the second largest biotech company in the state. I would define my role as the current steward of the FilmArray platform. As I look around this room right now, I see many amazing colleagues, current and past, and it's an honor to share the stage with Dr. Poritz, who is also one of the inventors of this platform. And it's my job today to continue the great development that all those inventors before me brought. So just by way of introduction, in terms of our newest developments, the system that Dr. Blaschke talked about in the last session that she helped contribute to has now grown to tens of thousands of instruments throughout the world. And today we produce tens of millions of reagent pouches to help detect diseases like respiratory, bloodstream, gastrointestinal, meningitis, joint infections, pneumonia, all throughout the world. So we are present in more than 45 countries. bioMérieux as a whole has more than 14,000 employees, and I would say that the molecular range is really the most important part of the business today. So we are so excited this year that we can announce the launch and the FDA approval of our newest instrument and system called the BioFire Spotfire system. From ideation through manufacturing, this is a product of Utah. This was started in Utah, and it's produced here as well, the reagents and the instrument. It was a $125 million investment over 7 years. COVID really interrupted our timeline, as it did for everyone, I think. But this is the latest system in our molecular sample-to-answer platform. It's fast. So our previous platform was an hour. This is now a 15-minute time to result. It's comprehensive. We can do our same 30 to 45 target multiplexing sample to answer on this instrument that we can do on the rest of the FilmArray today. It's scalable, so you can run from 1 to 4 modules. The module's about the footprint of a piece of paper, and then you scale high from there. It is incredibly easy to use. It takes about 2 minutes to load a test, and it's so easy my kid can do it. And it is very connected. We have bidirectional connectivity, the latest STIGs for cybersecurity, and we want to have LAS connectivity as well so that the information can get out of the box to help inform the patient. This has been approved by the US FDA for both in-hospital and CLIA-waived use. We have 2 panels on the market today, a respiratory panel with up to 15 targets of organisms or organism families, and we also have a mini version, the R-Mini, that tests for 5 targets. It's just a subset of the full respiratory panel. So we're pleased to bring additional advances into the point of care space, as you'll hear about today, with our syndromic multiplex testing. So.
Very good, thank you. I'm glad you have a smart kid that can actually run your instrumentation. That's really good.
I didn't claim that he's smart. He can do it, though.
All right, very good. Let's go next to Mark Poritz.
Hi, I'm a molecular biologist and CSO of Co-diagnostics. Before that, I was at BioFire, and as we've talked about, I worked on the development with Kirk Ryrie, the founder of BioFire, and one of the things I can now claim credit for is hiring Andy Hemmert. But it was easy because at the time Kirk was in school and taking a chemistry class, and Andy was the TA and said, "You got to hire this guy." So it was a no-brainer. But do you have my? It's a PDF actually. Yeah. Next slide, please. There. So Co-Dx is a public company, about 10 years old. We grew dramatically during the pandemic, sold 35 million tests under an EUA. It was founded on an unusual flavor of primers that, if they're designed carefully, dramatically reduce the amount of primer dimer and some of the background signal, and it can increase the annealing temperature and speed up the PCR. And the CEO of the time, the CEO of Co-diagnostics during the pandemic said, we need to think about the next thing. So he first initially contracted with Idaho Molecular, which was formed by Kirk Ryrie and Carl Whitwer, who had left BioFire and University of Utah at that time. And so we started that in 2021, and then were acquired, and now, so it's got quite a few employees formerly at BioFire and and other parts of the valley. Next slide, please. So, sorry, I do this, okay. So we are developing the Co-Dx PCR Pro. It's a platform for over-the-counter and point-of-care testing. It's meant to give you reverse transcription quantitative PCR results in a very inexpensive, easy-to-use system. So our anticipated price for the disposable will be $20. For the instrument will be $300. There's no setup or calibration required. It's manufactured in Utah, and we're really aiming for ease of manufacture so we can scale it up dramatically as needed. As you can see, there's a small device that's not much larger than in height than a cell phone, and we use the cell phone as the user interface. And in fact, we've tried to reduce electronics in the device by having it talk by Bluetooth to the cell phone, and the cell phone talks to the cloud where all the data is stored. So before you start a run, you scan the instrument to connect to the instrument, scan a barcode, you scan the barcode on the disposable, that information is sent to the cloud, it checks that it hasn't expired, that disposable hasn't expired, it's not recalled, that the right protocol is being run. And then at the end of the run, the user comes back with the cell phone, uploads the data, the analysis is done there, and that result can be emailed to the patient or to the healthcare provider, and as appropriate, to public health. So we're really trying to reduce electronics and outsource it to the cell phone and to the cloud. And we're close to completing, we have completed the clinical trials for coronavirus test that we'll be submitting for an EUA to the FDA shortly. We have in development an upper respiratory panel that will be flu A, flu B, RSV, and coronavirus. And then we've received, initially it was $1 million each from the Gates Foundation for MTB test and a human papilloma high-risk variant test. And they just recently, we announced that they've given us now additional $9 million to scale up the manufacture of them and get approvals for the TB test and to start manufacturing parts of that in India. So we're aiming for small multiplexes, and with the right sample type, we think that the value proposition is in the cost and the speed together.
Okay, thank you very much. So you can see a broad array of tests are actually represented by the companies on this panel, people with a lot of experience. What I thought we would do is spend some time diving into a set of questions, and I'll add a couple more to this as we get into it. The first question to the panelists is this: what do you find is the most challenging aspect of developing diagnostic products? And I'll give a list here. It could be the discovery process itself, right? It could be the underpinning technology that you choose to develop. It could be regulatory strategy and uncertainty. It could be identification of a market opportunity. It could be actually reimbursement strategy and execution. And it could be the education that's required of customers, and that includes physicians, patients, healthcare workers, and payers. It could be any of these things. But what have you found to be particularly challenging? And if you feel comfortable, share some war stories with the audience. Okay? Who'd like to grab that one first?
I'll grab it.
Please.
So I just got back from DC last night, and so it's no surprise that the FDA rulemaking on laboratory-developed tests is probably the most challenging thing that I've dealt with in my career. So as many of you probably know, the VALID Act was not passed by Congress, and the FDA decided to proceed through rulemaking for laboratory-developed tests, and they are using the IVD framework, and they are saying that laboratory-developed tests are in fact a device, and that if it's a high-risk device, it's going to need to go through a PMA. If it's not a high-risk device and doesn't meet this 1976 exclusion, it's going to have to go through a 510K-like process. So there are no exclusions, there is no grandfathering, and it's open for comment right now. And comments are due by December 4th. And this sounds like a pitch, right? And so I, encourage all of you to read this lovely 83-page document and put your comments out there. But no, seriously, we see this. There's a lot of assertions in this document that we think are not true, including that 50% of all LDTs are problematic. And so it's kind of painting the whole industry as being, bad players, bad tests, and that's just not the case. This is likely to go into effect by April of next year. And even though I anticipate there will be, lawsuits, definitely from the American Clinical Laboratory Association, of which ARUP and other large laboratories are a part, that means that the rule has to go out first before it can be challenged. So I see this as a big threat to clinical laboratories everywhere, including academic laboratories that bring up their local tests. There's many tests that we offer. For example, we do a rapid whole genome sequencing for sick babies in the neonatal ICU. We have results back in less than 7 days, and we have diagnostic answers in 40 to 50% of these infants. This is an LDT. There is no FDA-approved test. Given the choices that we're going to have to make as a shop with a lot of LDTs, we can't submit them all to the FDA. It's as you know, it's a really expensive process, and it's going to take a lot of time. And I don't think the FDA really knows what they're biting off here. So that's one test that we would likely have to pull off the market. So it's a big challenge.
Okay, so the uncertainties with regard to regulation are certainly a problem. And let me just ask this question, and I know the answer. It's going to be an obvious answer, and I'll let you answer it. Talk about the validations that you go through in general before you put a test out there and use it clinically. Because I think this is one of the things. There are some bad players out there that actually throw things out that are not properly validated, and it paints the entire industry with a bad brush.
Yeah, so we have really extensive validations. I'm sure that many of the companies up here, where we're looking at sensitivity and specificity and precision and interferences and and so on. And we're often doing this as part of large clinical trials as well, because in order to, depending on the population that you're looking at, right? This takes us year to 2 years. If we're doing a companion diagnostic, which is FDA regulated, it could take us up to 7 years, and we have examples of that. We have the first companion diagnostic for gene therapy that's available in the world, and we currently offer it right now. So it's a very rigorous process, and we do it right. Is it going to be what FDA wants? Probably. But we're going to have to go through that submission process. So we're worried. We don't think FDA can handle this. It's estimated that for them to handle what are potentially hundreds of thousands of LDTs that would be submitted, that they would need up to 33,000 regulatory folks. Have you tried to hire these people? Where are they? I'd like to know, because they're really hard to find. So even with third parties, like with deemed status, like maybe Wadsworth, New York, maybe even CAP, it's not enough to handle the volume that's going to come.
Okay, thank you.
Mind if I jump in on
Please
that just
do.
a little bit?
Yeah.
Having worked with ARUP, where we would develop a test, it would go through that rigorous process of sensitivity, specificity, precision, and then to pass it on to someone else like ARUP, and then have them go through that entire process again on their end, ensuring it's like a double. It only strengthens the power of that LDT, in my opinion.
Yeah, and the one other point that I'll just have people think about, and by the way, I'm not biased one way or another. I think that there are a lot of pitfalls having the FDA step in and regulate on the one hand. On the other hand, there are some benefits too. It's like all things. There are pluses and minuses. But the pace of innovation has been cited as one thing. It will be slowed by this process. We won't be able to have new diagnostic tests developed as quickly as possible, and that's one thing that people are concerned about. So Tracy has no strong feelings about this, and she made it clear. The people that submit and go through the FDA process, for many of them, it's a process they're very familiar with. There are many diagnostic tests that actually receive FDA approval or clearance, and that happens. It's a matter, of course, for many people, but in some cases it's not. So it's an interesting question. We will not resolve it here, but we will move on to a couple of other issues. Okay, so. Yeah, please do.
Another answer. So I would phrase it slightly differently, that with modern diagnostics as represented here, often the systems are complicated enough that you have engineers, molecular biologists, manufacturing, regulatory, and what happens is you have a tendency to horse trade the risk. So given the choice, the engineers would say, you don't mind if I boil your enzymes while I'm inserting them into the instrument. And we say, no, that probably is not going to do very well. And so usually it's much more subtle than that. But my point is that it's easy. What can be hard is just figuring out who got left holding the bag when the most technically complicated thing was given to someone, you just didn't realize it, and they have to deal with it.
Yeah. Good point. Okay, there will be more on this perhaps in the halls. The next question I have is, we heard a lot about AI today, okay? And so I'll ask this question. What is the impact of AI, machine learning, and other technologies on diagnostic development in your hands or in the hands of close colleagues that you have?
Yeah, so I'm happy to talk about how we've been able to use machine learning and AI to actually improve our tests. So we have a really nice feature with the film array system called Syndromic Trends, which Dr. Ports was also a part of creating, in which we're able to partner with many of our customers, have their data anonymized and uploaded into the cloud, and we're able to see in real time what pathogens are trending across the United States. It's a really incredible website. I encourage you to look at it just to see the different patterns in respiratory pathogens, for example, over time. But we also learn a lot about the performance of our assays and our tests through that process. And just as an example, we noticed in our past that we had one of our assays begin to drift through our Syndromic Trend software, and we're able to use the machine learning information to help signal to us that we needed to make an improvement on that assay very quickly. Because as we know, these viruses mutate and change over time. So it was able to help us be very responsive to what was happening across the US in real time and be able to adapt our test. So it was so incredible to have all of that data. And then even now, it's been amazing to watch through the pandemic and afterwards, through those data, the change in circulation of respiratory pathogens. We have yet to see a real flu season, and who knows if it'll come this year or not. So, pretty cool to check out.
Okay, good. Any other participants? Yeah, please.
So I think this is something that's touching on every aspect, from the very early development all the way through what you're able to do with the data once you have results. I think it's also an area, though, that's been overpromised and underdelivered by our industry. We have been talking for decades now, at least for as long as, and I haven't been in the industry for decades, but decade and a half or so, we've been talking that entire time about data, and what we're going to be able to do with it, and all of the decisions that we're going to be able to be more effective and more accurate in making as we begin to pool this data. And I think one of the realizations is that the quality of the data that we put into our algorithms, whatever they may be, is critical to turning out a result that actually matters. And so I think that's a great example of a use of data. But I think as we move closer to the point of care and we start to have geotagged information, this begins to open up additional areas. So when I look at Co-diagnostics, or I look at my own company, or Q, or some of the others that are playing truly in that point of care space and are working as hard as they can to get into the homes, this begins to give you really accurate, pinpointed information of how disease is spreading in real time. And I think only time will tell what we're able to do with that. But I gave an example at an event we held last week where we were working with the Atmospheric Research Institute, and they were looking at trends with weather patterns and the spread of disease. And one of the things that they found, granted post, is they found that with every 1 degree centigrade we increased with global warming, we saw a 5-fold increase in the spread of dengue around the dengue belt. And that becomes really powerful information once you have immediate point of care information of where an outbreak is. And as you're able to have that in real time, all of a sudden there begins to be a suite of tools that we don't even think about. Things like mosquito abatement around that area in order to try and stop the spread of the disease. So when we look at this, yes, there's impact for patients just generally. There's impact for us as we're doing development because we are able to develop and improve our technology so much faster with this new suite of tools. But then I also believe there's a new generation beyond that, where as the data continues to improve, I don't know what all will come from it, but I think it's going to be incredible.
Thank you. Other thoughts? Yes.
Yeah. One thing that I think about a lot being in the immunoassay manufacturing business is the quality of the antibodies that we use. Really, it comes down to often we can only build an assay that is as good as the antibodies that go into it. And I'm seeing papers come out where AI is being used to tweak and tailor these antibodies in a way that really has never been possible before. And we're seeing improved affinities, improved avidities, improved stabilities, and I'm so excited for that to trickle down through the thousands and tens of thousands of antibodies that are out there so that we can start to find those antibodies and put them into our products to make that even better.
Very good. Stay tuned for more. Okay, I'm going to change the question a bit. Talk about the ability for you to find and to retain talent. Each of you runs a company that's trying to do something that when you're creating new diagnostics, you're trying to create something that doesn't really exist. You have to have people with special skills. Tell me how you find them. Tell me how difficult it is to find them. Tell me about what it's like being in Utah and trying to innovate. And where do you go to find your talent? How do you get there? Anybody?
Yeah, I'll jump in. First of all, let me just say thank you to the other panelists because we've recruited out of their organizations, and I say that with actual true sincerity. Yeah, it's funny, but the cross-pollination that we have in this ecosystem right now, we've grown to a point to where we're finally starting to see true cross-pollination company to company. And what comes from that, in my personal opinion, is just an increase in the standard and an increase in the level that we're able to operate as companies in Utah. You guys saw some of the report, or we all saw some of the report that Kelvin presented this morning about this growing life science ecosystem that we have here in the state, and truly specifically in this valley. And that becomes really powerful and impactful as we continue to innovate. I look at companies like Recursion, for example, and multiple companies have been started because of team members there. That's of great benefit. And so in terms of recruiting, I'm grateful for the other companies that are here on this stage as well as here at this conference because they are training up people who have come now and worked for us. And we have also had people leave and go work in other places. Now, having said that, there's the positive. The negative is we just don't have as large of a talent pool as we need. And this continues to be incredibly difficult. We spend a fair amount of time recruiting. In fact, I think 4 of the last 5 hires into our company were out of state, and we brought them here. And luckily, again, there we have an ecosystem that at least we can tell a story of. This is a great place to live and to work with a really high quality of life. And then in terms of like retaining talent, I really believe that companies have to think differently about what individuals mean. And there's a new generation of team members with a new generation of ideals, and finding people who align with your organization on a very deep level, where their personal beliefs and the beliefs of their company align, that creates a really high level of retention.
Very good. One question that's sort of related. How many of you are letting a large number of your employees work remotely? Yeah.
So we have a policy in our organization of 50% hybrid, and then it's also somewhat team dependent and function dependent. There's certain groups where to be competitive, you have to be full remote. There's other groups to where to do your job, you have to be in a laboratory. So it's a lot of, it depends on the group within the company, but we also do have a policy of that hybrid. Then we'll see how long we keep it.
Does that allow you to cast your net more broadly and actually recruit over the world?
For those groups that have that full approval to where we need to do it to be competitive, yes, we can look more broadly.
Okay.
We do the same thing. Is if you're working in the lab, that's 24 hours a day, 7 days a week, you got to be there, right? But then there's certain team members who, they're pretty hard to hire, and they want to move out of state, and so we've let them do that, and it's worked out really well. It depends on the person, it depends on the team and what their role is. And then, Utah's a great place to live, I'm just going to say. And so I've been really successful in recruiting people out of California from, friends and colleagues who I've used to work with, and they love it here.
Yes,
One
please.
thing we've noticed that for the remote people, if we're hiring new people and they're coming from out of state, that our HR manager is very reluctant to hire people remote from certain states because the burden it puts on us to meet their standards has been almost impossible. So we don't want to touch anyone from California, unfortunately.
Okay, I'm going to end with 2 quick questions. This first is a leadership question. What's your biggest challenge as a diagnostics development leader, as being the leader in this area? And then the last question is, what are the most exciting things that you have worked on or are working on? Anybody could take that, please.
I'll answer the 2 really quickly. The most challenging aspects is actually balancing the science with the people, with the politics, with the budget, with the timelines. It's a juggling act. And as a scientist by nature, I'm most comfortable with the science, but that actually is my own personal struggle of needing to do those things. In terms of the most innovative thing, I showed it already, Spotfire. We're so excited for what this brings to the world.
Okay. Please. So
I would agree with you about the juggling act. And I would say, one of the most exciting products that we've put out was a joint project that we worked on with TechSite, which is a startup out of Utah. And we put together what's called a stool tool, which, yes, that's stool, where we use machine learning and artificial intelligence in order to find ova and parasites much faster on a slide than a technologist could do, right? They'd spend many minutes, under the microscope. This machine learning and scanning identifies those organisms right away. It's better than a human at finding them. It's faster. It displays the result in a better way, and it improves our efficiency. And so we're now working without TechSite, but we're doing other tools like this within the laboratory to improve efficiency.
Jared?
Yeah, I'll also agree on juggling. I'll add to it as a startup, challenging is raising capital in a really complicated market. and a market that coming off of COVID is not valuing diagnostics the way they have been valued historically. And so that creates a very complex situation from a money-raising situation. From something we're absolutely so excited about, it is our Secret system, which again goes back maybe to some of the complicated things. It's actually really difficult to fight for innovation as a startup. The push is to try and get a product across the finish line and into the marketplace, and we fought for a really long time and to eliminate a power cord from an at-home diagnostic system that was running a full molecular process. And so it was years of failure and failure and failure and failure before we were able to find new ways of doing things that could be implemented into our technology, and I don't think we could be more excited about it, frankly.
Kyle?
Yeah, I think one of the challenges that we've found is actually just obscurity. Being a small biotech company in Utah, it's hard to actually find and connect with the right people, the people that are going to be able to use the tools that we make, and just balance, again, like everyone else has said, a juggling act of finding that balance of getting out there and finding the right people that will be able to use our tools and make those connections.
Okay, Mark, challenges and best thing you've worked on?
I would echo what was said before, but I would also add that there's a whole suite of new technologies, whether it's Teams meetings or OneNote or all the different ways you can, and I don't think we fully understand how to use them correctly, and there are sort of personal issues of making sure we don't talk over each other or have monologues during meetings. That's been difficult to work out, and we're getting better, but it doesn't get the attention it deserves to really make a tech company function efficiently. One example is just, no one has stacks of paper on their desk, but they also can't find things. So they email me and say, you sent me that document could you send me again? And I'd say that what's been fun is, a traditional thing in diagnostics is you measure your LOD with a contrived sample. You say, I can measure this many. And our initial data was not the strongest. And so RADx, which ended up funding us, was very critical of that aspect. But when we did our clinical trial, we are as sensitive as some of the gold standard commercial tests. So you really, how you use it, maybe it's because we're running it right there with the patient versus freezing and shipping off for reference lab testing. For whatever reason, it's fun to see how things work better than you think, and that's very nice.
Very good. I think that concludes our time. I'd like to thank our panelists, and thank you for your attention today.